Welcome to Brain Ponderings, your window into the world of neuroscience with unfiltered conversations with experts in the field. Uh, Today, my guest is Professor Stephan Sigrist, and he's going to talk about a fascinating molecule that's emerging as a very important player in aging in general, in, in brain aging, in neuroplasticity, and that it's actually a molecule in our diet. So I'm not going to tell you what it is yet. I'll keep you waiting. So Stefan is a professor of genetics in the Department of Biology, Chemistry, and Pharmacy at the Free University of Berlin. He's does a lot of work using Drosophila, the fruit fly models, because of the powerful genetics that can be used in them. He's made major contributions to the current state of understanding of the proteins and the interactions of proteins that regulate synaptic plasticity, particularly on the presynaptic side, the release of neurotransmitter, which is increasingly recognized as being very important. There's all this, not controversy, but in the field, it's been a lot of focus on the postsynaptic side in terms of plasticity, but the presynaptic side. So welcome, Stefan.
Yeah, thank you so much, Mark, for this very nice introduction and obviously for having me here today. It's a great fun.
Yeah, I'm looking forward to this. Okay, can you talk about your background? So you're from Germany?
I'm from Germany. I was actually born in the very southwest of Germany, a city called Freiburg, to then find my way to Berlin for my school time, West Berlin. I then started chemistry to begin with, got a little bored, I'd say, with chemistry, changed into biochemistry, which meant returning to the southwest to a university town called Tübingen. And this is then when I entered into biomedical research. And as you already said, I mean, like making the fly my pet organism. And that is then my PhD thesis, which is at the Max Planck there in Tübingen, where we looked into cell cycle regulation. You know, this were the golden days of cell cycle regulation. All these molecules like cyclins, etc., were identified. And we could use fly genetics in order to identify machinery, which was important for the degradation of cyclins. It's a protein called Fizzy and Fizzy related, which turns out to be degrading cyclins at important steps, so to say, of the cell cycle. And this is how I came into the fly. To then leave from there, obviously more direction neuroscience. I always kept the fly with me, so to say, as you already said, for its beautiful, wonderful genetics. Also for the fact now that we have a complete, I mean, electron microscopic map of the fly brain, just other papers published in Nature a couple of days ago. So it's just a wonderful organism, you know, to go from really molecular scale questions into all the way to behavior and so on. This is what is
Yeah,
so very
there's so many
interesting to us.
advantages, and sometimes people, the lay public doesn't realize the power of some of these invertebrate systems, particularly the fly and the worm. And our brains are so complicated, it's really hard to get at the core mechanisms and down to the molecular level. And for aging too, that's actually
Right.
of value because they have a relatively short lifespan. And humans, you can't really do
Right.
aging per se and look at lifespan, right?
Yeah.
You have to do biomarkers. So
Right. Exactly. So I mean, even applies. Let me say, I mean, it's a little challenging. I mean, because we're still talking tens of days for them to age. I mean, yeah. But I mean, obviously, in a PhD thesis, you can ask a couple of questions. I mean, if we're talking mice, in one thesis, you can ask exactly one question at best, and
yeah.
so on. So that's why we're doing both, I should say. I also have a very good postdoc, Marta, Italian. She does mouse work in the lab, but you better start with some hypotheses, you know, which you have been fostering in the fly, and then you can selected things you can test in the mouse. That's kind of our strategy, or even going into human trials, which we're part of in the past as well, with the molecule whose name we're still gonna tell the people. Yeah.
Okay. So before we get to this fascinating molecule, just a little
Yeah.
bit more of some of the work. So I had Tom Sudoff on one of the podcasts quite a
Mhm.
while ago, maybe a year ago, and
Right.
he got a Nobel Prize for his work on looking at proteins involved in regulation of neurotransmitter release from the vesicles outside. And you worked on that too, but you've added a lot to that. Can you talk a little bit about your contributions there?
Absolutely, my pleasure. Obviously, Tom's contributions are have been funding this field to a good degree. And yes, we do work on the very least machinery on the one hand, which is these famous snare protein, synaptotagmin, and so on, all these molecules Tom and some coworkers found and described. But this whole release process takes place in a complex nano environment, which we refer to as the active zone. And that's quite a bunch of additional proteins, actually, which allow this system to operate at this incredible speed. Also, some of our
Uh-huh.
neurons fire hundreds of action potentials. In a second. And this little synaptic release sites, these presynaptic sites have to deal with that. So there's incredible challenges in terms of like recruiting new synaptic vesicles, getting rid of the ones which just fused, pumping transmitter into the ones which have been just back endocytosed and all that. And that has enforced the evolution of a really complex nano architecture. And there is a good handful, or two handfuls, say, of such active zone proteins, which are to our advantage, are very nicely conserved, as we say, into the fly. And we have been describing their roles over the years, a lot of super resolution light microscopy, because, when you subject a synaptic active zone to normal resolution light microscopy, fluorescence microscopy at a confocal microscope, you're just going to see a little boring dot because we are just below the diffraction limit. And it was my time, actually, when I was still in Göttingen 2 decades back, that we got in touch with Stephan Hell, later Nobel Prize winner, on super resolution light microscopy. And so we were between the very first people who could use these microscopes, and we started to see that we can really describe these nano scale architectures, I should say, with these super resolution techniques. And we've been doing this over many years now. And so we see there is delicate architectures which do exactly the things which must be there. Like retrieve vesicles at high speed, so to say, get everything in place, exact spatial relations between the calcium channels and the release machinery. You need that for proper control of synapses, and do maintain quality of vesicles over time. Attract mitochondria. There's this, what I read yesterday, a little bit in preparation of our encounter today, is that it takes 20,000 ATP molecules in order to fuse and endocytose a single synaptic vesicle. So you can see, what energy demand locally is there, and also mitochondria are part of that engine, becomes clearer and clearer. And yeah, this takes us busy. And then also these structures, as you just said, there we see are very important, actually, in order to do also behavioral reprogramming of every kind. Be it learning and memory, which we can do in a fly brain structure, which is called a mushroom body, which is in a way the equivalent of the mammalian hippocampus. And then we also see, and that is actually something very important for us, is that sleep, and particularly sleep deprivation, so giving a bad night of sleep to the flies, is changing their presynaptic active zone architecture.
Huh.
And we think this is happening, or can also show to a degree, in order for the brain to come up with the rebound sleep you need.
Hmm.
It looks like there's a plastic process which is favoring, so to say, the transmission of neuronal activity patterns which execute sleep, so to say. And this is also pretty much how we got ultimately into the direction we're going to talk about today, because these changes of the presynaptic active zone in broader regions of the fly brain, we not only encountered in flies which are sleep deprived, we also found in flies which are, so to say, of advanced age. And so we more and more consider these plastic changes as a kind of like a generic, if you wish, resilience module of the fly brain, which can call for sleep and can also do benefit, we think, for in the aging process. And this is how we got into all that, and this is how we started to also work in direction of aging, essentially, fly brain aging.
On the flies, with the genetics, you have some funny names for genes and proteins
Yeah,
based on the phenotype of the fly. There's things like Sonic Hedgehog, for example, and Frizzled
yeah,
and
yeah, yeah.
all of this. And so can you briefly mention, before we get into the main part of our conversation, the
Right.
main topic that is, uh, so I looked at a recent paper you had. It actually just came out in Nature Communications on a protein called blobby.
Right, exactly. Yeah, exactly.
So how was that named, and who named it, and what did you find about its
Right,
role?
right, yeah. Yeah. Yeah, good point. Why this bizarre name? So, we're still in search actually of additional factors actually to execute control at these active zones. And in this particular case, we went into a biochemical approach where we used synaptosome preparations from fly brain. So you can harvest fly brain material by a procedure where you essentially freeze the flies and in liquid nitrogen, then you shake them on a vortex kind of, then you can sieve out heads. And so you can do biochemistry. I mean, it's like, because I mean,
You,
obviously
you
food flies
need a
are very
lot of
small.
flies. We did
You
some
need a
work
lot of
on
flies. It
synaptosomes
might
from.
be it might be bad for my karma one day, but yes, you need a lot of flies for that. You need, but it's still doable. I mean, like say you need a tray of these vials, of flies, and then we can do biochemistry of the kind also like Tom Südhoff and people have been using them actually a lot in mammals. And for example, preparing synaptosome, so biochemical fraction, which is rich in synapses. And on those, we executed a procedure called cross-linking proteomics, where you look for physically associated proteins. And we started, so to say, from our bona fide active zone proteins and looked for proteins to physically, so to say, interact with those. And we came across a new factor, which is, as many of these proteins, is large and it's having a signature of being most of it being intrinsically disordered. And however, to its end terminus, it has a rep domain, a rep gap domain, sorry, I should say. So something switching off particularly little reps, GTPases, which are also of importance for the active zone regulation. And then the usual thing in the fly is that once you have such a candidate, you go into genetics. And so we did genetics and then took sort of removed the factor and then looked into our synaptic terminals with their active zones. And we found these big blobs. So there was the material, which is normally meant to end up very neatly in these active zones, formed like a big, huge accumulation with a blob, so to say. And turns out that protein is very important actually to likely harvest material. There's continuous transport of active zone precursor material, so vesicles decorated with these active zone proteins. And blobby, as we then called it, travels with these vesicles, obviously. And then it's needed essentially to kind of like allow a controlled consumption of active zone proteins into newly forming active zones, particularly newly forming active zones, but equally active zones which are under active remodeling in order to, for example, form a memory. Because we see that when we only when we let the flies develop normally and then in their adult life, once they have formed their synapses, only reduce the amount of that protein by a factor of 2 or something like that, going to 50% of it by RNA interference, in particularly those neurons of the mushroom body forming memories, then we see that the animals are no longer able to form any longer lasting memory. So it completely eliminates memory consolidation. And yeah, now we're very curious to understand of what exact nature these remodelings are. We do see blobby is also of importance for sleep. Ultimately, we think these active zone remodelings, be it in the context of sleep deprivation, be it in an aging brain, there is quite some commonality. I mean, also when it comes to the formation of memories. And this is what we want to deeper understand,
If
essentially.
if you did you increase blobby levels and see if that somehow enhances learning and memory or counteracts the effects of aging or sleep deprivation?
That's a very good point. And we're not quite we not there yet. We have engineered lines, which allow us to overexpress from the endogenous locus by a CRISPR-Cas9 technique. And we're just starting to do that. And I should say, how I don't complicate too much, but we have also phenotypes of that factor if it comes to sensory decoding, these synapses. When we talk learning and memory in the fly, we usually refer to olfactory memory. So these flies learn a certain odor is bad, so to say, because it has been associated by the evil experimentator with like an electric shock, for example. And once they went through such an experience, they're going to avoid this odor later on. And we do know this is to do on the one hand with these neurons in the mushroom body to encode memories. On the other hand, you have to convey the olfactory information about the exact nature of this odor. And there also the active zone is of very importance. And how do I put this in a nutshell? I mean, there's different release machinery, so to say, within individual active zones. So there's some release machinery, so to say, which is looking more into kind of phasic information, so which is, so to say, when you have a complex pattern of action potentials coming in, which has different frequency components, something regular and then little bursts and so on. There's some machinery which is dedicated to look into this tonic, so to say, information, and some is more sensitive towards like the onset when there's a burst coming and so on. And we do see that Blobby seems to tune that equilibrium. Also, so in the sensory decoding, you do need an acute regulation of these release components. And this is, so to say, in the neurons before the mushroom body called projection neurons. This is the ones which carry the olfactory information into our brains. And there we're also having a very, very close, relation to the organizational design in mammals. So this whole olfactory system and the the glomerular structure of the first relay stage, we call it the antennal lobe. It's called olfactory cortex then in mammals and so on, but it's very similar essentially by design.
Yeah,
so
the
and I should say
chemosensation.
overexpression of Blobby is changing things there. Sorry, that's where I came from. Overexpression of Blobby is changing things. So we think it's not only a permissive thing, we think it might well be an instructive signal, but it's early days.
Okay, fascinating. So all proteins have amino acids have a NH2 group, the nitrogen, two hydrogens. So proteins have a N terminus where the NH2 is and a C terminus. So now you can introduce the molecule that has it's related to amino acid. Can you talk about polyamines and what is a polyamine and which one you've been looking at together with your colleagues?
Exactly, my pleasure. I mean, so the molecule we're going to talk about the most today, its name is Spermidine, and as Mark just says, it is in this group of polyamines. Polyamines means a couple of these amine groups we just talked about, which usually with standard pH accept a proton, so they become positively charged. And so this is very, very simple substances. So the most simple of them is a molecule called putrescine, which if you have a little bit of chemical education, you could also call 1,4-diaminobutane. So it's nothing else than a C4 body, linear chain, an amino group to the one and one to the other end. And from there on, nature then starts to, so to say, add additional hydrocarbon chains with amino groups. So when we put like another C3 amino body on that, ending up with 3 nitrogens, now 3 amino groups, we call it spermidine. If we do that another time, we call it spermin. Super simple substances known since the late 1800s, most likely pleiotropic in nature in that they bind many, negatively charged biomolecules. DNA, RNA, they're meant to be, so to say, important for the fundamental integrity. Lipids, which are also negatively charged often. And that's definitely a fundamental role of them, and not the least the name spermidine comes from the fact that spermidine, that substance is very, very high in sperm for the fact that the DNA has to be compacted very, very much in this sperm head, and then you find very high levels of spermidine. But don't get confused. I mean, these substances are there throughout our body. We find them in all tissues. And I think an important thing to say, spermidine, and we're going to talk about its beneficial effects today, or what we all hope to see there, is a body endogenous substance. So yeah, we have it, our cells produce it all the time, though somewhat to a dwindling extent with age. And our gut microflora, so to say, produces spermidines in different amounts. And then there is dietary sources for spermidine. But at first glance, it's simple and body endogenous bioorganic molecules, positively charged, which derive exactly from amino acids. So there is the so-called urea cycle, which is a major metabolic module which deals essentially with the degradation of amino acids, if you wish. And there is a substance called ornithine in there. And when we decarboxylate ornithine, when we take away CO2 moiety from ornithine, we arrive at this before mentioned putrescine. Sorry, putrescine. And from putrescine, we can go spermidine and spermin finally. And it's been already decades ago, it's been sort of observed that these ornithine decarboxylase, the enzyme which is controlling polyamine synthesis, is an extremely controlled enzyme. So when I studied biochemistry, we use it as a substrate for the so-called N-end rule. You know, the identity of the amino acid at the end terminus somehow controlling the lifetime of a protein. And very different from standard enzymes. Ornithine decarboxylase has an in vivo lifetime of only minutes, which is already a sign that there's some very active regulation. And then there's all layers of regulation on that you can think of. There is an antizyme which blocks ornithine decarboxylase. Then there's an anti-antizyme which blocks the antizyme and so on. Then there's all levels of transcriptional and particularly also translational control, which made people thinking since a while that maybe the exact levels of these polyamines in our body matter for a reason because they're so very well controlled. Yeah.
Now, amino acids there's transporter proteins in the membrane of cells that actively move amino acids into the cell, so when you eat protein and it's broken down to amino acids, gets in your blood, it's moved into the cells. How do the polyamines get into cells?
Right. I mean, that's a very important but still unresolved question.
Ah,
So, but there is good candidates now. There is this SLC3 family. Likely, there's a colleague of me, Peter van der Leuwe, in Belgium. They are characterizing these transporters in the moment.
ah.
There's one interesting transporter already described better, which goes by the name ATP13A2, which is a gene, and that gets us a little bit in a certain direction already, which is a gene which has been found in familial forms of Parkinson, actually, Parkinsonism, to be mutated. And turns out that here we have actually an endocytic uptake of polyamines by certain pits in the plasma membrane, which then leads over to
Ah.
the polyamines being present in our lysosomes. And this is where this transporter molecule comes into play and then allows for an exit of polyamines from the lysosomes into the cytoplasm and to some degree likely also into the mitochondria later on. That's
So maybe
subsequent.
less specific than amino acid. The positive charge of the polyamine may interact
Right.
with negative charge of the lipids in the membrane or something like that, and then that
Yeah,
triggers.
but I think Peter and colleagues they consider these transporters to be well specific. I mean, it's a good point whether they're
Okay.
all going to discriminate between spermidine and spermin and so on. Likely not, but I think it's a distinct class likely of transporters.
Okay.
But as said, this is really lagging behind characterizing these transporters. It's pretty clear that there is active transport. People can measure that to a good degree. It can penetrate the blood-brain barrier, though it takes some time for that, but it does. There's been isotope labeled spermidine just been injected, and you
Ah.
can find it then in the brain. You find it way faster, I should say, however, in heart and kidney, for example. But the very molecular identity of the transporters and to work out their redundant scheme of action. Likely there's a couple of these candidate molecules, maybe they have overlapping action, which makes it not too easy then with genetics and so on. That is still in the making, unfortunately. So we'll have a Gordon conference on polyamines. So I'm been have lucky to be the co-chair of this meeting, and we're gonna hear more about transporters. I think it's a very hot topic. People have neglected that. Now with the increasing interest in this family of substances, we'll better understand, I think, in short time.
Okay, so then are there, so I quickly look back at some of the literature from plant plant science,
Right.
because
Mm,
plants produce these polyamines and and
mm,
and they produce them in response to stress,
right.
various types of whatever, I don't know, heat or salt, or
Exactly. Yeah.
they can increase a lot in the plants.
Right.
Um, so we can increase our spermidine levels through the diet. I guess there's certain plants that may have more than others or something like that. Or I guess a supplement. I see a lot of people like in
Eating this.
the
Yeah.
popular culture are buying
Yeah.
spermidine, but it seems like you can just eat something with the spermidine.
Yeah, you can. I mean, so what I personally eat is wheat germ, and it's overall it's like that. Plant embryos are very rich actually in it. And I should say then there's maybe for some people consider maybe more tasty versions. Is there's some mushrooms. There's also some fermenting cheeses which are very rich. Not all of them. Broccoli, for example, beans, peas, nuts. There is a Japanese variant of it, which is called natto, and I'm not sure you've
Ah,
heard about
I've
that.
had fermented soybeans. Yeah.
Fermented soybeans. I don't know how you liked it. I mean, it's not mine, I must say.
Well,
Love that,
I put it on like some vegetables or something, but
right?
the reason I took it is not spermidine, it's vitamin
Yeah,
K2.
right. Exactly. It's rich in various things.
Yeah.
Exactly. It has this reputation in Japan to be very healthy, and there is a literature actually which shows that by natto consumption you can increase your serum levels of polyamine. Already, kind of like 2 decades back, people were interested in that. I must say, I have a hard time to make it part of my diet. I love Japanese cuisine in general, but definitely, but not natto. I think it's
Yeah.
an acquired taste.
Well, well,
It has to be your grandmother serving it to you then.
the wheat germ's good. You can put that on a lot of your
Right.
cereal or whatever.
I do a muesli from wheat germ, and I think it tastes very well. I like that. And as you said, there's all these food supplements on the market now. I'm afraid not all of them necessarily contain as much spermidine as they claim. I mean, that's a little disclaimer here, from what people told me.
Now there's very little or no quality control on
Little
these
more control.
because
Yeah.
they're supplements and they don't have
Yeah.
to, they're not regulated very well.
Right. Yeah. Exactly. Yeah. That's yeah.
Okay,
Um,
so let's get into aging.
right.
And so Frank Matteo, actually I've never met him, but our the things we worked on over the years have overlapped, and Rafa
Mm,
de Cabo, who was a good
right.
friend of mine, and actually he was a postdoc in my laboratory a long time ago, and
Mm,
then he's a
mm,
lab chief at NIH doing a lot of work on basic mechanisms of aging and other ways you can increase lifespan in mice. And they're not, the one we know about is caloric restriction or intermittent fasting,
right.
powerful effects.
Mm.
Can you talk a little bit about, and I can't remember whether you were on this Nature Cell Biology paper in 2009 or not, but
No, not.
um, well, yeah,
But
it had flies in it, so
that's
probably,
lies in it, and honestly, I should say Frank and me were old friends actually from studying biochemistry, and so no, but I couldn't convince anybody in my lab those days to really do it. But I was certainly very impressed by this paper, and then we got into that question of brain aging in the fly, and whether we could find anything to preserve memories in aging fruit flies, because also fruit flies, when they age, when they are kind of like mid-age, which would be 30 days, roughly 4 weeks of age, they would start to effectively form new memories. And we thought, can we do something about that? And then first of all, we used dietary restriction actually, and it would just not, which would just prolong their lifetime a lot, but would just not do anything to the memory scores. It would still decay with pretty much the same kinetics.
hmm.
And so this is then when we looked into Frank's paper, and I spoke to Frank, and then we tried on spermidine actually, and this is how the thing started for us. Yeah, and this is sort of like 2010, 11 or something like that. And we subjected the fruit flies then by just putting 1 or 5 millimolar spermidine into their food actually, and measured memory function across their aging trajectory, so to say, and found that when at an age of 30, 40 days, where usually they do no longer form a specific form, I should say, of memory by this mushroom body formation we mentioned before, this would in normal animals just no longer happen. And in fruit flies under this spermidine supplementation, that would still happen with a high efficacy. And that, I mean, there is a huge interest from us, I mean, certainly to understand how that intersects with aging and to help to make this a candidate, I mean, to be tested further, I mean, including human trials in the end. On the other hand, we're also fundamental scientists and we saw, okay, we have a brain now. We did something, I mean, not too complex to it. We gave it 1 millimolar of
Yeah.
spermidine, and once a sudden it still forms memory. So how about the synapses? I mean, what's really changing? Can we dissect out? I mean, that in more detail. This is what kept us busy also to the very day to good degree. This is how we got into that, so to say, then in the early 2010s.
And the studies, well, Frank, there's a lot of people involved in those studies on the papers, but essentially they kind of provided evidence that the effect of the spermidine in increasing the mouse lifespan was not due to causing like a reduction in energy intake. It's doing uh, something. There must be maybe there's some convergence somewhere.
Yeah.
But I think that must be the big question now is there convergence and exactly what is the convergence?
Yeah, yeah. So we think of two processes essentially. I mean, the one is autophagy, and the other one is mitochondria and age-associated changes of mitochondria, which you can, so to say, really influence in a positive manner by keeping the spermidine levels high. I should maybe start by saying, I think we didn't mention that so far. I mean that there's a literature saying that spermidine levels decline in many organisms with age, including the human being. We looked into that in flies. We saw it declining in fly brains actually with age in that time window, so concomitant, so to say, with the memory scores declining. This was the very first thing we did essentially. And then in our first papers, we tested actually whether, in fact, inspired by Frank Madeo's work, whether it would need autophagy induction actually in order to explain the effects. And yes, we saw that there was autophagy induction. We saw that substrate proteins, waiting to be degraded by autophagy. For example, this Ref2, we call it P62, Ref2P, there's different names for it. It's an adapter molecule which sequesters, so to say, autophagic substrates, usually accumulating in old brains, no longer there at that level under spermidine diet. So yes, there was signs that autophagy was sort of like boosted in these aging fruit-fly brains, and also genetically we showed when we also did subtle things, when we would take away a single gene copy, for example, of autophagy regulators like ADG7, that already would occlude the spermidine effects. This was our standing, so to say, in the very beginning. Always having certainly the problem, how do we really connect now from the levels of this kind of innocent little molecule to the levels of autophagy and so on. What we do think though, and this is a paper of us in the early 2020s, 21, I think, is that a particular specific process, which is called hyposination, is at least a very fundamental or like important contribution to the whole thing. And if you wanted, I could say
Yeah,
a few.
talk because I wasn't
Yeah,
aware that hypocyanation. I'm aware of acetylation and anyway,
right.
this is a modification of a particular amino acid lysine, which is one
Exactly.
of the 20 amino acids.
Exactly. Yeah. I mean, there is very interesting reports also of acetylation, deacetylation equilibria to change, as work by Guido Krömer, for example, having identified a certain deacetylase, which might be target for spermidine to be activated and so on. But yes, but on the other hand, then it's been known. I mean, that I learned when I first so to say, visited this Gordon conference on polyamines. I was surprised there was a Gordon conference on polyamines, and I came there, and there was these people had found like a modification of a single lysine in the whole proteome. And it's the amino acid number 50 actually in the translation initiation factor 5A to be modified in that is an aminobutyl group transferred to that particular lysine group, and then subsequently there's a hydroxylation. So aminobutyl group put on a single lysine of this translation factor.
Wow.
The enzymatic system to do that, two enzymes which operate in sequence, the deoxyhyposine synthase, first one, then this hydroxylase, conserved from yeast to man like histones. I mean, incredibly conserved thingy. And this hyposine has been identified as an amino acid modification in the 80s, and then people have looked for a long time, found these enzymes. Then some people thought, I mean, okay, I mean, enzyme conserved, I mean, all the way. I mean, there must be more, targets for that modification. Looked for more substrate, didn't find any. It looks really like an enzymatic system evolved only to modify this lysine. I should now say this modification, the donor of this amino butyl group to be transferred on this lysine is spermidine. So this enzyme deoxyhyposine synthase takes spermidine and transfers these aminobutyl group to spermidine and followed by this hydroxylation event.
And
What's interesting about these enzymes already is that when you are losing a single gene copy, you are having problems. I mean, like you can clearly show that also in flies, in zebrafish, for example, in mice. There's human patients also which suffer like a reduction in the activity of these enzymes. So it's very dose sensitive. And then we started actually to check out whether this was of any importance for the effects we saw in fruit flies, in terms of aging, but also very much in terms of our memory protection. And we did saw that when we would feed spermidine to the flies, that their hyposination level, to what degree this translation initiation factor is hyposinated, would increase. And we could also show that when we would remove a single gene copy of these enzymes or also point mutate this particular lysine in this translation initiation factor from a lysine into an arginine, then the flies would become somewhat short-lived. I mean, and particularly they would no longer be susceptible to spermidine supplementation. They wouldn't care anymore. And
so,
I should maybe
Stephan,
say,
Stephan,
sorry,
is
yeah.
is is spermidine in stimulating protein synthesis or inhibiting protein synthesis?
Right, a very, very good point. We think it's qualitatively changing the spectrum of what you're going to translate.
Ah,
And so what we observed was that when you did proteomics of these fly brains, but also similarly when Frank Matteo and colleagues looked into yeast cells, you might have overall more actually a shutdown of some translation, yes, but you do clearly have a boost, for example, of mitochondrial components. And this is also going along with the functional status of mitochondria being boosted in aging tissue by spermidine supplementation in a hyposination-dependent manner. Mark, if you allow me, just one sentence on what this thing really does. I mean, how comes this translational, this little prosthetic group they put on this translation factor plays such a role? And the thing is, I should say, it's a misnomer. It's likely not a translation initiation factor. It's likely an early elongation factor. And there is open reading frames evolved, which often have nests of glycines and prolines, where it gets very hard for the translation elongation machinery to read through. And only if you had the initiation factor 5A being hyposinated, it binds into the P side of the ribosome and, so to say, clicks it further, promotes effective translation. And it looks like there's a set of mitochondrial protein, there's likely a set of autophagic proteins, which in their initial open reading frames have these little nests of glycine proline, which makes them susceptible, so to say, to the polyamine concentration, because only the hyphosenated form would promote synthesis then. And this is our best explanation in the moment why we get this reprogramming. So there being really a shift of the mitochondria into a way more juvenile state, so to say, under spermidine supplementation. And that has been found in flies and C. elegans, also very convincingly, I'd say, in mouse brains, actually, that this effect.
So spermidine increases autophagy,
Yeah.
autophagy,
Yeah.
and is it specifically then, based on what you just said, promoting the removal of dysfunctional mitochondria by mitophagy?
That's a very good point. I mean, well, we think, I mean, we've seen that not only mitochondria turnover is increasing, but it also is that cytoplasmic substrates, like the ones I mentioned now by this piece, sequestered by this P62. I think both is
Okay.
served by the induction of autophagy, which in aging tissue you see upon spermidine supplementation. Which of the ones is the most important? It's hard to tell. What we follow the idea in the moment, is that this spermidine elevation might be a signal which serves two connected things same time. So yes, there's good data. It's boosting mitophagy, so it might be responsible for taking away overaged mitochondria, but particular in the periphery there of the neurons.
Yeah.
But on the other hand, what we do see, it also helps the biogenesis of mitochondria by, and we suspect local translation actually at close to the synapses of mitochondria. So we have some unpublished work which makes us believe, so to say, that there might be a spermidine controlled, polyamine controlled local translation of mitochondrial components in a cytoplasm in order to be imported into mitochondria. And there's
Yeah.
also, to add up, sorry, there's a boost also of the in mitochondria translation, we think, because we find mitochondrial translation machinery to be high upregulated under spermidine supplementation on proteomics level. Mechanisms are still to be worked out. I mean,
Okay, so mTOR, right? So what
right.
the things you kind of mentioned, is it's well known that mTOR pathway is involved in uptake of nutrients, particularly amino acids,
Mm,
and when it's active, there's a lot of just kind of global protein synthesis increases in cells. If one inhibits mTOR with rapamycin or by fasting, actually,
yeah,
that will enhance autophagy. So there's a kind of a negative connection between mTOR and autophagy.
right,
Is there a role for does spermidine have any effect on the mTOR pathway?
right. That's a very important question. It is that I'd say if I survey the literature, which has been looking, into spermidine supplementation in different aging organ systems, the overwhelming body of work which has looked into that would say there is no straight direct
Okay.
connection here. With, I should be fair, a few exceptions, which have seen modulations of TOR activity. There is this recent paper actually on fasting and the role of elevations of spermidine levels upon fasting. And in a nutshell, what they show in yeast is that when you reprogram yeast in order to undergo into a starvation period, so to say, they say that in order to switch TOR off, you needed an elevation of spermidine, because they report, I mean, upon like in a fasting yeast cell, so to say, you'd have this increase of spermidine synthesis, and that seems to be a precondition how direct we'll have to see in order to allow the switching off of TOR to happen, and thereby to switch the whole system also from a more like glycolytic into a more oxidative phosphorylation state. But the mechanistic details are not there. They're not there, and it has to be urgently to be worked on that.
Okay, so this hypocyanation of the EIF5A translation elongation factor. Um, so is that is that lysine subject to acetylation and deacetylation or ubiquitination, which is another
Right.
modification that can occur on lysines and that's related to autophagy?
Absolutely. I mean, there is even reports of the 5A itself being subject to acetylation, deacetylation. And so it might be that these mechanisms intersect even on the level of the 5A potentially. You might acetylate it in order to not allow it to be hypocynated, or we'll
Right.
see. There's reports of that kind. In terms, I mean, there is a lot of crosstalk likely to acetylation under spermidine supplementation, be it just for the fact, that when you start to supplement spermidine, you likely trigger the flux through the system, means you're also going to degrade some of these supplementary dietary spermidine, which you do by acetylation followed by an oxidation reaction. So you might start to deplete your acetyl-CoA pool actually in favor of degrading this excess spermidine which you got, and
Hmm.
so there might well be a sort of interaction on this level. And as said, I mean, there is reports actually of more like a deacetylation state upon spermidine application in yeast, and that's been shown very clearly, and that might also then even sort of intersect with epigenetic reprogramming and histone modification potentially. So I'd say so. There's lots of reason to think that these systems might interact on such a level. In terms of ubiquitination and the whole UPS system and so on, not so much so far, I would say. From what
Okay.
I've seen, we have a little uh, re research group here funded by the German Research Council. So a dear colleague of mine, Janine Kirstein, she's looking into exactly that question in the moment.
Yeah.
But that's where I should stop, or where I think it's too early to report. It's been looked into, but so far no real positive evidence, I'd say.
Are you familiar with, so what's going on with people in, doing models, various models of Parkinson's? You mentioned Parkinson's
Right.
or
Yeah.
Alzheimer's, and
Yeah.
there's relevant models in flies and mice.
Yeah. Right.
Um, yeah, so
Yeah.
what's going on there with spermidine?
Right. I mean, there is let me start like that. I mean, like saying that the relation we had mentioned this ATP 13A2, this transporter. I mean, so there is in the Parkinson's field, there is kind of like a causal relation in a
Yeah.
way between misregulation of polyamines and neurodegeneration. Is there already based on these kind of findings?
And
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 there
and mitochondria and
And then
ubiquitination.
because mitochondria
Okay.
might suffer because the lysosomes are no longer able to effectively export their polyamines into the cytoplasm. And that might subsequently affect the mitochondria. And you do know mitochondria are meant to be a major convergence point
Yeah.
for Parkinson's, and not the least with Pink and Parkin and so on, and mitophagy not being effective anymore. Which might be another inroad into that. And per se, to be frank, I mean, the relation between polyamine, so to say, level changes and the progression of neurodegeneration is a little complex, in that maybe there might be periods also where brain tissue might, it's been reports on that, starts to increase polyamine levels. And this is the usual problem you're having with all that. It's not easy to separate,
Ah, yeah.
some things which are real drivers of the aging process and of the pathology versus things which are attempts to rescue and so on. And so this is in the making. So we here in Berlin could contribute a little bit to a collaboration with colleagues in neuropathology, and they looked into mouse models of A beta, and they would find that the A beta load in mice under spermidine supplementation or diet would reduce. There's also data on that being of cognitive effect, and they hooked it up though to also the action of microglia. So microglia being more
Hmm.
effective in maybe removing this A beta plaque debris.
Hmm.
Um, then there is early work also again from Frank Madeo's lab on fly models and even yeast models of Parkinson kind of pathology, which also reported like positive effects. And I do know that there's a lot of serious, say, Alzheimer's mice models, particularly being tested in the moment. But again, I think in order to robustly say whether we can expect something, it's again a little early days, and these things take long time because also the mice have to age, etc. All that. I mean, so we'll see. But I think there's reason for optimism, and the question is, ultimately, if you thought about spermidine being a dietary recommendation one day, we certainly have to face, I mean, that the whole sequence of Alzheimer's, we're talking decades likely. So it would be more a recommendation likely for people in their 30s, 40s. So I'm
Yeah.
afraid we both are too old for that
Yeah.
in order to stop the process. And there might be a point of no return. And so it's very hard to tell. I mean, yeah, but yeah,
Okay.
people are trying.
And in relation, so there's a
Mhm.
lot of evidence emerging from people working on intermittent fasting and caloric restriction that, so it turns out all of the early work on caloric restriction in rodents, the way the experiments were done, the animals are actually fasting for up to 20 hours every day,
Mhm.
because then the restricted group, they give them their daily allotment of food all at once, say 30 to 40% less than they normally eat.
Mhm.
And when they get that much less food, they eat it all right away.
Mhm.
And so there's this metabolic switch that occurs from, using glucose as an energy source to using ketones, so
Right,
ketogenesis.
right, right. Mhm.
And there's a lot of evidence now that many of the beneficial effects that were initially attributed to caloric restriction per se are actually due to metabolic switching and ketosis. And one of the ketones, beta hydroxybutyrate, has signaling function.
Yeah.
Is there any relationship at all? I didn't see anything
Very good
on
question.
any relationship between
Right.
spermidine and ketones.
Yeah, I mean, I could easily so think of that because of, as we said before, the acetyl-CoA cool pools maybe being, there might be a metabolic sort of like interaction between the two. But on the other hand, I also lately looked into that, browsing literature, didn't find much, used my contacts, not the least, I should say, Frank Madeo, phoning him, asking him that question, and no, not so far. Well, I mean, it might be a win-win in the sense also that if ketone bodies use another mechanistic inroad actually into their, I think, very robust beneficial effects, the better. I mean, then we could combine things. I mean, yeah.
Yeah, or the spermidine could be like
Yeah,
permissive for some of the effects of beta hydroxybutyrate, the ketone, on gene
right.
expression. There's also some evidence, although I guess it's controversial, that there can be a modification of lysines in proteins by beta hydroxybutyrate. It's called
I,
hydroxybutyrylation.
I see. Okay.
So yeah, there's some potential connections. And so
Yeah.
you mentioned very early on that these polyamines are positively charged. They interact with
Right.
DNA, and so you could imagine and there's evidence that the ketones affect chromatin,
Right.
right? And so there
Yeah, no,
could be
definitely.
some interaction at the level of chromatin and the essentially the exposure of genes so that they can
Yeah.
uh, be
Yeah, very
transcribed.
much. I mean, I think that, there is this in this early work of Frank Madeo, actually, they essentially found, I mean, that there is epigenetic changes in the sense of a denser packing actually of major parts of the chromatin likely in by taking away some of the histone modifications. And that leads overall to like less transcription if you wish, but in favor of selected packages of genes. I mean, this is early work in yeast. And this is this is what I know in terms of like how could there be a bridge to epigenetic effects, knowing obviously that ketone bodies have a clear connection there. And as said, I mean, I could imagine that the acetyl-CoA pools are kind of modified by spermidine, and many of these acetylases, their Km is in the range that they might be reactive to level changes of acetyl.
So I know what I'd do if I was
Yeah,
going to do experiments. So you got the
yeah,
flies, and you can reduce endogenous spermidine by RNA interference,
with ornithine decarboxylase, for
the
example.
pathway, and then
Yeah,
so I guess first thing is seeing is there an effect of the ketone beta hydroxybutyrate on something
mhm,
in the flies related to
yeah,
plasticity or gene
yeah,
expression, and then then the question would be if you reduce spermidine levels, is the ketone no longer able to uh, have these
yeah.
effects or something like that.
Sounds great. Sounds great.
Yeah.
You want to do it? All right.
Well, I
Tell me.
retired from the NIH
Yeah,
5 years ago, so I
yeah,
can be a consultant, an unpaid consultant, and if something comes out of it, you can
yeah.
put me in the acknowledgments.
Oh, well, more than that, but and it sounds great. I mean, we should definitely talk about that. It's very interesting direction. I do have two like very motivated new students actually, which just enter into
I'll
our
send you some papers on the.
great. Yeah, yeah, would be my great pleasure. Yeah,
I've got a kind of a perspective article in that's under review for what is it for Nature Metabolism on essentially on the important role of metabolic switching. The idea is that it's important to subject your cells and organ systems to intermittent bioenergetic challenges to optimize health. So it's well known with exercise.
yeah,
Same thing with intermittent fasting. So then the question, what's going on there? And so the idea is you get these cycles of challenge period where you cells go into a stress resistance, conserve resources mode.
yeah,
Autophagy goes up, for example, mTOR down,
right.
growth and plasticity are kind of suppressed. Then
Yeah,
during the recovery period, eating, sleeping, resting, the cells go into a growth and plasticity mode where things like mitochondrial biogenesis occur. So the
mhm,
switching back and forth over time.
yeah, yeah,
Uh,
absolutely. Yeah, yeah.
yeah.
And- and-
So there's a role for the ketone in that. So that's
yeah.
sort of point.
Also, as an inter-organ communication system, I mean, or like, I mean, is the liver the pacemaker of all that ultimately, or
Yeah, well, that's where the ketones are produced. So
yeah,
probably, but yeah, I consider the ketone as hormones,
right, exactly. Yeah,
and there are
yeah,
other hormones that are circulating. Yeah, but that's the whole nother question, right? These
right.
intercellular. Well, that gets the question, do different cells in the brain, are they capable of producing different amounts of spermidine and under different conditions?
Yeah,
Like, what if you stimulate neurons?
right.
Will they increase spermidine
I mean,
production?
there is reports also on spermidine being secreted, be it from neurons, be it even from glia. I mean, glia seems to have higher levels of polyamines even. So, but this is all these only superficially easy questions, like I
Yeah,
don't have to
well,
tell
yeah,
you that measuring the free
but
amount of
you've got to start with superficially easy questions to
yeah,
design, doable experiments. So
right.
it's
Yeah,
good to,
exactly, exactly.
right.
But there is a little light on the horizon concerning measuring levels because there is now more kind of
Oh,
fluorescent sensors to
wow.
report about the free concentrations of these polyamine spermidine, which have been so to say published just lately. I mean, so there was great stuff on this polyamine conference in Kobe last year, actually, and so there's a little bit hope that we're going to
Okay,
be
so
able
there
to
may be
monitor.
some fluorescence indicator of spermidine levels that you
Right,
can
exactly, exactly
actually
that.
image the fly brain and
Right, exactly. That's
whatever.
yeah, yeah. Image the fly brain and also under starvation, for example, to see whether this
Yeah.
is
Yeah.
the pulse of polyamine, whether and then you could genetically engineer the fly in a way that you restrict where so to say biosynthesis can happen, and you can relate that to well, the behavioral readouts and the sheer fitness and longevity and so on. Yeah, that's
Okay.
yeah,
That's
yeah.
a fascinating area. A lot to do from the basic science standpoint and clinical. Do you know about, you may know about, are there human clinical trials of spermidine in any disease conditions or other?
Right. I mean, yeah, there's quite a lot ongoing. So we here in Berlin actually, based off these initial results we had from flies, we got some support from the Federal Union there in Germany, and had a consortium which also included clinicians, colleague of mine, Agnes Flöhl, and they conducted a first, I'd say still pilot screen, if you wish. I mean, like, so people complaining about subjective cognitive decline in their 60s and 70s. And then 100 people being recruited, which is tough work, I must say. And then 50 of them on the placebo, 50 with these wheat germ extract, which is between us, been a very low concentration, likely, because there's been technical limitations of sort of like concentrating spermidine in a food supplement compatible manner to a degree that you could swallow it still as a tablet. I mean, it's better now. Now we can give them like 8 milligrams a day easily in one tablet. In the old days when we did this screen, we were down to something like 2 milligrams, which is very little. And then there was memory testing, like hippocampus dependent memory things the neurologists emphasize you just cannot train. I mean, you can really look into sheer memory performance. And when we did a subgroup analysis, I should say, in the people in their 70s, not in the ones in their 60s, but the people in their 70s, where we could also measure over this 1 year of study, indeed, in the placebo group, a certain decline. There was actually a trend, at least, in direction of beneficial effects on memory function. And this has now been a couple of years that we published that. And what we learned from that is we need higher concentrations. We need at least, say, at the same amount you take by dietary habits, just that. I mean, double your input. I think that's fair. I mean, that would still make us arrive below the level Southern Italians anyway, consume every day. And do that. And then also we should certainly look into people who really in that window of intervention would have a high chance of undergoing a cognitive decline. Yeah. And so now we are in close contact to an institute here in Berlin, which is a nutrition specialist, and they have access to people in their 70s and also 80s. And we've just been running a trial where at least where we all we did was essentially ask the question: is 3 weeks of like something like 8 milligrams spermidine a day, is that sufficient to boost your autophagy just based on the parameters we can take, which is blood, obviously? And yes, I mean, there in PBMCs, we did see a really robust boosting of autophagy, which is, again, I mean, like just kind of like a little precondition to get into more serious clinical testing. This is talking about what did we do here, and then there's a lot of stuff ongoing. I mean, there's people looking into also other centers looking into cognition and age-induced memory impairment. There is studies ongoing also in the north of Germany, Rostock, where this colleague of mine, Agnes Flöhl, the neurologist now is. She looks particularly into sleep, because sort of say difficulties of sleep are so closely associated with neurodegeneration that it's very hard to tell who is egg, who is hen, and so on. But on the other hand, it's a nicely quantifiable readout. Sleep deficits usually kick in already in the advanced mid ages, I can tell. So, and this is being used as a readout ongoing in the moment. And then there is a still unpublished study, but in bioRxiv, which is led by Niels Gassen, a colleague from Bonn, also in Germany, And they actually looked into depression. And they would argue that depression is kind of like a stress state also.
Yeah.
And that for example, if you prepare for there being a stress period, and this bioarchive is very funny, starts with a bungee jumping cohort. So they look into people who are going to face a bungee jump, and they say, I mean, there's an increase of your autophagy levels, I mean, and polyamines actually, in that in order to prepare you for likely pre-stressful. And what then Niels and people did were to actually administer Spermidine, I mean, like into people who had never seen antidepressants, so freshly diagnosed people. And at least these bioarchive papers, I mean, should, with caution, I mean, it's not peer reviewed yet, I mean, but sees like an efficacy which is at eye level with established medicamentation there. And they say they see also an increase of polyamine levels in those subjects which under standard pharmacological diet have a good prognosis or developing well. As if you have to kick off, so to say, your polyamine elevation as kind of a stress response which gets you back actually into normal performance. This is what this study says. I think sometimes it's too good to believe. I know, but I think it's doing something fundamental in the end, in reprogramming of aspects of brain metabolism. I mean, so maybe it's not so surprising in the end that it has this broad spectrum of action. And so and I don't have to tell you what a big problem depression is. And if just we could do a dietary or food supplementation recommendation to take for individuals which are endangered and so on, that would be great already. Yeah.
Well, does physical exercise affect spermidine levels?
Is being looked at. Again, a friend of mine, Simon Sede in Graz, is looking into exactly that very systematically. So he got a very generous grant actually there from the county of Steiermark there in Graz, and Simon looks into exactly that. But
That's a very powerful antidepressant exercise.
yeah, good point. Good point.
Yeah.
Yeah, absolutely. And it could well be that it works via spermidine, or it just converges onto the same things like mitochondria and so on. I mean, that
You can do that in the flies.
you
Put
can
them
do
on
that
a
in
treadmill and
yeah. That's a good point. And again, I tell you, I mean, people do all kind of freaky paradigms on the fly. That's been done. I mean, yeah. We tried a couple of years back. We're not so successful. I have
oh,
a talented postdoc with me. She wants for her own future. She wants to go this direction. It's actually kind of like a treadmill enforced locomotion. You can also do this optogenetically or thermogenetically. You can engineer the flies, in these locomotion centers, and it's a few command
ah,
neurons, and so you can make them just walk with the light, so to say. I mean, to exercise if you wish. Yeah.
interesting.
And, and yes, I mean that should be doable. Yeah.
Okay. All right, Stefan. I enjoyed meeting you and chatting with you. Thanks for
Yeah.
taking the time. This is an
Yeah.
exciting area, and right now, when I'm done talking to you, I'm gonna order some wheat germ and
Okay.
combine that with my oatmeal. I guess most whole grains have
Whole grain
spermidine.
has it. Yeah. Yeah. Exactly.
Yeah.
Exactly. So
Okay.
oatmeal should have a lot of it already. I mean, yeah.
Okay.
Yeah. Yeah.
Good.
Mark, it's great fun. Thank you. Thank you so very much. Yeah. Yeah. Yeah.
Okay.
So
Bye.
take care. Take care. Bye bye to everybody.